Several sources can contribute to bias in a testing system. Calibrator or reagent lot changes can be a source of both imprecision and bias. When the shift is sudden, notable, and persistent, then it's generally due to bias. Recalibration can also be a source of both imprecision and bias. Recalibrations are an opportunity to introduce new or additional incremental bias. Recalibrating more often than recommended by the manufacturer can point to a problem in the test system, or due to a process control system that's inappropriate for the test and is producing false error flags. Another source of bias is off-brand reagents, calibrators, or consumables. These can be substandard to manufacturer ones. For example, a cuvette provided by the manufacturer could have more consistent optical quality than an off-brand cuvette. Reagents that are not stored at the proper temperature or are stored in direct sunlight can degrade. This can cause bias. Tests that are sensitive to technique can show bias when there's a permanent change in operator. An instrument that reads or interprets the calibration curve differently after a software upgrade can cause bias. An instrument can show bias when manufacturer instructions for maintenance are not followed or are followed poorly. Participation in interlaboratory comparison and external quality assurance programs can help a laboratory identify issues with bias and imprecision. Performing routine and proper maintenance on instruments, controlling environmental factors, consistent operator technique, and proper reagent storage are just some of the ways laboratories can control analytical variation. Let's review what we've learned about analytical variation. Analytical variation is the variation that occurs during the analysis of a sample. There are two types of analytical variation: random imprecision and systematic bias. Random variation is a measurement of ongoing randomness in a test system. Systematic variation is a sustained variation where measurement and direction are relatively persistent over a period. Tests that are susceptible to imprecision are those requiring manual steps or are technique sensitive. Sources of imprecision are instrumentation, environment, operators, and the age, stability of reagents. Methods of measuring imprecision: repeat testing of QC materials and controlled experiments. Both shifts and trends can be an expression of bias. A shift is a sudden and persistent increase or decrease in QC values. A trend is a gradual increase or decrease in QC values that eventually stabilizes. There are two types of bias: true and comparative or relative. True bias is the difference between an observed value and the value for the same analyte derived by a reference method that's metrologically traceable to an international standard. Comparative or relative bias is the difference between an observed value and the calculated value of a consensus group for the same analyte. The peer group is the ideal and preferred group for statistical comparisons. It contains analyte data from laboratories using the same instrument, method, reagents, temperature, and unit of measure. The method group contains analyte data from laboratories using the same method, temperature, and unit of measure. Use this comparison group when the peer group is too small for reliable statistical comparison. The All Labs group contains analyte data from laboratories using the same temperature and unit of measure. This is the least statistically valid consensus group. Possible sources of bias: recalibrations and reagent lot changes, use of off-brand reagents, calibrators, and consumables, improper reagent storage, software changes, and poor or no instrument maintenance. For all of your laboratory QC needs, go to www.qcnet.com.